So, with that said, I would like to welcome our first speaker of this section, Roxane Chen, who is a PhD student at the University of Cambridge, and she's going to be sharing with us the overview of how resistance mechanisms happen in lung cancer. Roxane, the stage is yours.
So it really is a privilege to be here, and it's been a real opener to see all these amazing speakers and learn from all of you. So today I've been asked to give you an overview of resistance mechanisms in lung cancer. But before we dive into that, we first need to understand how the treatments work and therefore how cancer works. So we'll start with the normal cell. So this is very basic stuff for some of you here. But we have the cell here with the nucleus here and the DNA that codes for everything that your cell does. It gives out these instructions to the cell by producing RNA or messenger RNA that can then travel out of nucleus into the factory of the cell to create proteins. So because my PhD is on ALK-positive non-small cell lung cancer, I'll take some examples from ALK, but I promise I'll cover other things as well. So normally the receptor, non-mutated, just sits at the surface of the cell, not doing anything, until it gets activated by another bit of protein. And once it gets activated, the receptor gets phosphorylated. That means that there's a phosphate group that gets attached to it. So the phosphate gives it energy. You can remember that yellow P as being powered on. And so once it's powered on, it will tell the cell that it's time to grow. So usually cell growth is a very regulated process. It's like cars going through a toll booth. You have to make sure that the cell is healthy before it's allowed to proceed. But in cancer, usually due to an assault on the DNA, so we often think of smoking, but it could also be due to just bad luck. You get this mutation in the DNA that gets passed on to your RNA and then to your protein that is now also mutated. And it's mutated in such a way that it's always powered on. So it always has this phosphate group giving it energy. And now it's telling the cell that it's always time to grow. So this is like losing the brakes on your car, or if your foot is always on the gas pedal, and now your car is just speeding through the toll gates, and there's no checks on it, and it's just growing and growing and growing. So if we start with a normal clump of cells, one of them gets this mutation, and it starts growing much faster than the rest until eventually it forms this solid mass, which we call tumor. So you can think of a tumor as a house with the cancer cells living in it as, the people living in a house. So like in any other house, you need water and electricity to supply the people living in it. And so the cancer, or the tumor, does that by creating blood vessels to supply it with the things that it needs. And we call this angiogenesis. So we know that the cancer is actually an invader. It's not supposed to be there. So if someone just started coming into your garden and started building a house there, you think that your guard dogs or immune system would try to chase it away. But the cancer can be quite smart and actually recruit these guard dogs to work for it and protect it. And this is what we call the tumor immune microenvironment. And depending on where your cancer starts, you get different histological subtypes. So at the peripheries, it's usually adenocarcinoma, and the central airways, it's usually squamous cell carcinoma. So I start with tyrosine kinase inhibitors or targeted therapy because that's what I'm comfortable with. And again, taking the example from ALK, but it works the same way with EGFR. So ALK has a promoter gene that helps it, and they usually sit separately on chromosome 2. But due to mutation, they get stuck together, and this results in a fusion protein that is also always active, and this leads to cancer. So our generations of ALK TKIs all work in the same way. It binds to where that phosphate group giving it energy would usually bind and stops that signaling from happening. And the cancer cell dies without any of these growth signals because it needs these growth signals in order to survive. So resistance can arise. The more commonly known one is called on-target resistance. So that's when a mutation within the ALK protein stops the TKI from binding to it, and it gets activated again, and you have cell growth. The other kind is called off-target resistance. So here your TKI is working perfectly fine, it's still stopping ALK signaling, but you have many other receptors on the cell that can start signaling and become active. So MET or EGFR in ALK-positive lung cancer can cause this cell signaling again, bypassing ALK inhibition. So we call this bypass resistance mechanisms. And remember when I talked about histological subtypes, you can actually switch from one histological subtype to another, so from adenocarcinoma to squamous cell carcinoma. And this is kind of like your house switching from a wooden house to a brick house. you can still break down the brick house, but it's going to take a different kind of therapy. So moving on to another type of systemic therapy, chemotherapy. So chemotherapy targets all of these fast-growing cells. It's like a speed detector. And that means it also targets any cell in your body that grows quickly. So your hair and your intestines, which is why you see the side effects that you do. So chemotherapy essentially stops from going from one cell to two cells. It's trying to stop that from happening. And in order to do that, you first need to double the amount of DNA that you have by creating copies of it. So gemcitabine pretends to be one of the building blocks of DNA, so it gets integrated into it, and then the cell can't use it, and the cell dies. Carboplatin directly damages the DNA, and that's also what radiotherapy does, and again, the cell dies. Etoposide will stop the unwinding of that DNA. No replication, the cell dies. And microtubule agents like paclitaxel will physically stop the separation of your two bits of chromosomes. And usually with chemotherapy, we give it in combination. So there are two different ways of killing the cell. So carboplatin here at the top with pemetrexed. And so we're trying to maximize our chances of killing the cancer cell. However, they can become resistant to chemotherapy, so most of them cause damage to the DNA. But if your cell is just better at repairing that DNA damage, you're not going to see the effect of chemotherapy. It can also take up less of the drug, and once it's inside the drug, there are enzymes that can inactivate it, or it can get pumped out faster so that it doesn't have as much time to take effect. There are also other proteins in the cell that can tell the cell, you have to survive at all costs. I don't care, just do what you need to do, survive. And the last one is a phenotype change. So it's similar to the idea of a histological transformation where you switch from one house to another. This is the cell switching from a differentiated state, like a mature state, to something that's less mature, something like a cancer stem cell. And that will help it survive chemotherapy. So immunotherapy is a recent development in the field, and it uses your immune system to target these invaders. So we know that they're different from the rest of your body's normal cells, and they have these little bits of protein that your immune system should be able to recognize. And once your immune system can present that to your T cell or your white blood cell, and if it also has this activation signal, it can then expand and then go around your body trying to look for other bits of cancer that also presents this cancer antigen. And then the T cell that's activated can then attack and kill the cancer cell. However, your body is a very careful balance between go, go, go signal and stop signal. And the stop signals are always stronger than your go signals. So this is a safety measure to stop your body from attacking itself to prevent autoimmune diseases. So if it just so happens that your T cell also has one of these inhibition signals, it's going to be stronger than this activation, so you're not going to see that expansion. And your cancer cell can also present PD-L1, so programmed death ligand 1, which is also a stop signal, and it will stop the immune attack from happening. So what immunotherapy does is to stop the stop signals. So if you stop that inhibition from happening, you restore the activation of the T cell. And if you stop that don't kill me signal from happening, then the immune cell is able to kill the cancer cell again. So this is how the different immunotherapies work. Ideally, this is how it works in what we call an immune hot tumor. So this is a tumor that has a bunch of these T cells going around. It's able to recognize that bit of protein, and it's just exploding the cancer cells. This is like your guard dog that's really fit and healthy, has picked up the scent of that invader building a house in your garden, and next time it sees it, it's just attacking. But in immune cold tumor, your white blood cells are not invited to the party. They're not inside the tumor. The cells are in might be collaborating to help the tumor survive. And on top of that, not only they're not invited, but they're also completely exhausted. So they may express these markers of exhaustion that now researchers are trying to target to reverse that from happening. And so you can think of this as if your guard dog was in your neighbor's house and sleeping away and not able to do anything. On top of that, if this dog never learned to recognize that bit of protein, it just doesn't know who's the enemy and who's friendly, so it's not going to attack. And some cancers don't have a very specific antigen for your immune system to learn to recognize. And the last bit I'm going to say is angiogenesis is not great. So the blood vessels create this barrier that stops the immunotherapy from getting to it, which is why we give anti-angiogenics like bevacizumab to stop that blood vessel growth from happening. So I've given you a lot of information, but what you have to remember from this session, and I hope it sticks, is that cancer starts as cells that won't stop growing. Targeted therapy will stop those growth signals, and again, the cancer cell needs these growth signals in order to survive. Chemotherapy will target any fast-growing cells, which is why you see the side effect that you do. And immunotherapy enables the immune system to fight back. So thank you so much for your attention, and thank you for all the work that you're doing, raising awareness. So please keep advocating.